Hello and welcome to this series of modules on recognising and reporting tree bleeding. In this module, we're going to focus on some of the pests and pathogens. My name is Charles Lane, I work for Ferroscience Limited and I'm a Royal Society of Biology Senior Plant Health Professional, and I provide scientific advice to the Observatory Project. So in this first series of slides, we're going to focus on some of the phytophthora and fungal causes of bleeds. So as a plant health professional, when I think about bleeding trees, I always tend to think about Phytophthora as probably the most classic cause of a tree bleed. It produces these amazing water-soaked lesions, the very characteristic tar spots, and then underneath the bark you may see the classic foxy red-brown discoloration. In the bottom you can see some really nice examples of how Phytophthora ornei is causing that foxy brown discoloration and eventually will lead to the death of all these trees. So we're looking at a snake bark maple here. Here's the healthy bark, you can see green live tissue. Here's the leading edge or the junction between the healthy and the diseased tissue, and you can see the water-soaked necrotic lesion. What is also really nice is you can see a little bleed here. You can see further evidence of this as you go down the dead necrotic stem. But also what is quite interesting here, that this stem isn't showing any symptoms yet. The other thing I'd also encourage you to do is just pull away some of this leaf litter here and actually look at the root and the root plate to see whether you can see any necrosis or damage on those roots. So Phytophthora is more closely related to the water moulds and has a high reliance of water in its life cycle, so it's quite common to see Phytophthora infecting the roots and stem bases of trees. As you can see in the right hand slide, here is the discoloration you would expect to see underneath the bark of a root. On the left hand side you can actually see the classic tar spots further up the stem. So although Phytophthora prefers to infect the roots and the stem base, it can on occasions go quite high up the stem. So in this slide I've shown you two classic symptoms of a Phytophthora bleeding canker. On the right hand side is a close-up of the dark, dry, somewhat crusty black spots. They look like spots of tar, so not surprising are referred to as tar spots. On the left hand side we've taken a step back so you can see there's quite a number of spots on the tree. It is really interesting to look at the distribution of these tarry spots or bleeding cankers on stems of trees. It's really helpful to understand whether it's basal or aerial. How far has it travelled vertically up and down the trunk of the tree? It's also important to understand whether it has girdled the tree. If all those tarry spots coalesce together, they will kill all the sapwood underneath. That will then girdle the tree and result in a much more rapid death of the aerial parts of the tree. So you might want to record the number, you might want to record the size, you also might want to report the distribution of the spots around the tree, because it can be very helpful to monitor the progress of these spots on the tree to give you an indication of how active the infection is and the potential prognosis of the tree. In the next slide we've got some really good illustrations of Phytophthora bleeding cankers. We have two on beech and one on birch. Now these have been taken from the Observatory Guide on Phytophthora ramorum. Although this isn't a priority pest and disease, it is an organism we are concerned about, and therefore we have produced a really good illustrated guide which shows you some fantastic pictures of the range and diversity of bleeding cankers you might see on trees. We're now going to look at a true fungus that can cause bleeds on trees. Although it goes by the scientific name of Armillaria, you've probably more heard of it as honey fungus. And as you can see in the top right hand corner and beneath it, these are the honey coloured mushrooms or toadstools that you can see on heavily infected trees. But also importantly, the fungus produces water-soaked lesions at the stem base and on the root flares, which can be confused with other pathogens such as Phytophthora. Armillaria commonly girdles the base of the trees, causing the top of the tree to die. It's very rarely seen higher up the stem or in the branches. Now this can be a point of difference from Phytophthora. As we saw with the beech trees, you can get tar-like spot bleeds much higher up the stem. This wouldn't be expected to be seen with Armillaria. So in this slide we're seeing two very good diagnostic features of honey fungus caused by Armillaria. On the left hand side we're seeing the rhizomorphs, which are commonly called bootlaces. These are fine black strands of fungal mycelium that grow beneath the bark. On the right hand side you will see the creamy white mushroom smelling fan-like mycelium that grows underneath the bark. These are two key diagnostic features for honey fungus, and you would not expect to see these with other organisms such like Phytophthora. In this slide we're going to focus on Phytophthora lateralis, which is one of the observatory priority pests and diseases. It's an aggressive fungal-like pathogen that causes extensive damage mainly to Lawson cypress trees. The pathogen primarily attacks the roots of its hosts and forms lesions which extend up into the root collar and stem base. The disease is nearly always fatal and can have a major impact on trees in woodlands, windbreaks, parkland, private gardens and nurseries. The main symptom of this disease is the discoloration and death of the foliage in infected hosts. The pathogen produces lesions in the living bark layers of the tree, which eventually girdle stems and branches and deprive the host of water and nutrients. The foliage of infected hosts will initially turn a slightly lighter green colour than healthy counterparts, followed by a reddish-brown bronze colour as it dies. The colour change from light green to brown can occur rapidly. The lesions produced by this disease are tongue or flame-shaped with a sharp demarcation between the diseased tissue, which is a cinnamon brown colour, and healthy bark, which is a creamy white pink colour. Lesions are not visible unless the bark is removed, although resin bleeds, which we call resinosis, on the outer bark may indicate an underlying lesion, and this is what we're illustrating in the two pictures on the slide. In this slide we're going to look at the bleeds you might see associated with Psirococcus sugae, which again is another observatory priority pathogen. It's a fungal pathogen that causes shoot blight and cankers on cedar and shoot blight on hemlock. It can attack seedlings and saplings as well as mature trees. The pathogen has the potential to cause considerable damage to valuable ornamental trees in public and private gardens and economic losses for the forestry and nursery sectors in the UK. The most conspicuous symptom of this disease in the Atlas cedar is a pink coloration of the needles, followed by dieback of the affected shoots and partial shedding of the needles. Cankers can also be produced on the bark of shoots and branches, and in the picture you can see an illustration of a canker. The cankers can cause dieback and death of the shoots and branches when they become severe. Dieback in the side shoots can cause the shoots to have a bent over or shepherd crook appearance, which occurs as the shoot dies back from the tip. The cankers can be difficult to see as they are fairly indistinctive and observed only as slightly depressed areas of purplish black bark. Gum or resin production is often associated with these cankers, and small black fruiting bodies may also occur on the surface of cankers and on the dead needles. We're now going to look at a number of bacteria that can cause bleeds on trees. Acute oak decline, commonly called AOD, is a complex decline disease involving a number of bacterial species which cause bark lesions and a native oak jewel beetle. The disease causes a rapid decline in oak trees and can kill them within 5 or 6 years. Although AOD is mostly noticed on mature oaks, mainly 50 years or more, much younger trees can also be affected. The most obvious symptom is the oozing of a dark, sticky fluid from the cracks in the bark on the stem of affected oak trees. This stem bleeding usually occurs between 0.5 and 2 meters above ground level and can be visible around the entire girth of the tree and along the whole length of the stem, extending up into the canopy. The number of bleeds present on a tree can vary from just a few to up to 50 in a severely affected tree. The bleeds are vertically aligned on the stem and originate from longitudinal cracks between the bark plates caused by dead patches beneath the bark. These cracks are typically 5 to 10 centimeters long and have blackened, ragged edges. In some cases, the bark cracks heal and become occluded by callus material, and this becomes more visible when the outer bark around the affected areas has been displaced or fallen off. In the slide, you can see 3 different pictures. On the left-hand side, you can see a very characteristic ooze at a relatively low level. Remember I said that they would occur up to about 2 meters high. You can see the dark, sticky fluid oozing between the cracks in the bark. And someone once described this like flat Coca-Cola, which I always find a really helpful tip when looking for symptoms of AOD. And then finally, on the right-hand side, you may be very lucky, but you don't always necessarily have to see the D-shaped exit hole of the jewel beetle. So the bleeding usually occurs in the springtime and then again in the autumn, and it is very infrequent in midsummer or winter months. However, the dark sap stains the bark, leaving dry, black streaks on the stems, which may be visible all year round unless the rain washes them off. In this slide, we're going to look at some of the symptoms you might expect to see on a beech tree. We previously had talked about seeing some of the Phytophthora bleeds that you might see on a stem, but beech trees are also very susceptible to a Pseudomonas bacterial canker. In this slide, we're illustrating the rusty brown tar spots that you may see on the stem or trunk of a horse chestnut tree. Frequently this is a bacterial bleed, but there are also Phytophthora that may cause bleeding on trees. And in the left hand side you can see the dieback and premature loss on a horse chestnut tree. We're now going to look at one of the observatory pests that can be associated with tree bleeds. Great spruce bark beetle causes damage to spruce trees by tunneling into the bark of living trees. On hatching, the developing larvae excavate, feeding tunnels and galleries in the inner living bark layers. When beetle galleries completely girdle the stem, the part of the infested tree above the wound will ultimately die. This may take several years of sustained attack, with large breeding populations accumulating before individual trees are killed, creating a risk of spread to nearby trees. Resin tubes are characteristic of great spruce bark beetle infestations. These are produced as the female beetle bores into the bark to lay her eggs, causing the tree to respond by producing a strong resin flow in an attempt to protect itself. The resin tubes range in colour from creamy white to purple and brown. The presence of resin tubes accompanied by resin bleeds are useful indicators of beetle infestation. In this slide you can see the bleeding can be quite high up in the main stem, or it can be down at the base of the tree. And on the right hand side are some great pictures of a resin tube. In this slide there's further symptoms of the great spruce bark beetle. This is resin bleeding on a mature spruce, the characteristic resin dripping from beneath a branch, and then resin bleeding and frass at the base of a tree. So hopefully you found this second module useful, describing some of the symptoms you might expect to see from a range of different observatory priority pests and pathogens. Before we move on to the third module, I'd once again like to thank Anna, Susie, Charlotte and David for helping us bring this presentation together.